Hello students, today let us learn about growth media, environmental factors and explants source. Let us start with the introduction. The optimal growth and morphogenesis of the plant tissues cultured under sterile conditions in vitro is affected by many factors, and the response of the cultured tissue may vary for different plants according to their nutritional requirements and also depending upon the part of the tissue used as explant. The main factors affecting the response of in vitro cultured tissues are growth media, explant source and environmental factors. Let us first see the first factor, that is growth media. The growth media, or simply the culture media, is one of the most important factors affecting growth and morphogenesis of plant tissues, and the culture media supply the entire essential mineral ions required for in vitro growth and morphogenesis of plant tissues. Some frequently used basic media in plant tissue cultures include Murasige and Skoog or MS medium, Linsmeyer and Skoog or LS medium, Gamborg or B5 medium, and Niche and Niche or NN medium. These media formulations differ in the concentration of one or more nutrients, and a suitable medium is generally selected depending upon the requirement of the cultured tissue. Now let us see the composition of plant tissue culture media. A basic plant tissue culture media generally contains the following components, that is macronutrients, micronutrients, carbon and energy sources, vitamins and myoinositol, amino acids, undefined organic supplements, growth regulators, and solidifying agents. First, let us take up macronutrients. According to the International Association for Plant Physiology, the elements that are required in concentrations greater than 0.5 millimolar per liter are defined as macroelements, and they comprise at least 0.1% of the dry weight of plants. The macroelements used in plant cell or tissue culture media includes carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Nitrogen is required in greatest amount and is most commonly supplied as a mixture of nitrate ions from potassium nitrate and ammonium ions from ammonium nitrate, while phosphorus is usually supplied as phosphate ion of ammonium, sodium, and potassium salts. The second component is micronutrients. Microelements or minor elements are required by plants in concentrations less than 0.5 millimolar per liter and mainly include iron, manganese, zinc, boron, copper, and molybdenum, although cobalt, iodine, nickel, aluminium, sodium, and chlorine are also frequently present in some media formulations. Iron is usually the most critical of all the micronutrients, and is usually added as iron sulfate in conjunction with ethylene diamine tetraacetic acid or EDTA to form ethylene diamine tetraacetic acid or EDTA iron chelate, which gradually releases iron in the medium, making it available to the tissues. The third component is carbon and energy sources. Sucrose at a concentration of 2 to 5% is the most frequently used source of carbon in plant culture media. Other carbohydrates such as glucose, lactose, maltose, galactose and sorbitol are also used, and supplements like sugarcane molasses, banana extract and coconut water also serve as alternative sources of carbon for some media. The fourth component is vitamins and myoinositol. Vitamins most commonly used in plant cell and tissue culture media include thiamine or B1, nicotinic acid and pyridoxine or vitamin B6. Although other vitamins such as biotin, folic acid, tocopherol or vitamin E, pantothenic acid, ascorbic acid, riboflavin, P-aminobenzoic acid are also used in some cell culture media. Nicotinic acid is often added to culture media at a concentration of 0.1 to 5 mg per liter and pyridoxine and thiamine at 0.1 to 10 mg per liter. Myoinositol is a carbohydrate generally used in plant cell and tissue culture media at concentrations of 50 to 5000 mg per liter, along with the vitamins to stimulate cell growth of most plant species. The next component is amino acids. Although plants can synthesize most of the amino acids required for their optimal growth, certain amino acids like glycine and other amino acids like arginine, asparagine, aspartic acid, alanine, glutamic acid, glutamine and proline or amino acid mixtures such as casein hydrolysate and adenine are added in plant tissue culture media as sources of organic nitrogen in culture media, particularly for establishing cultures of cells and protoplasts. The fifth component are undefined organic supplements. Some organic extracts or natural substances such as protein hydrolysate, coconut milk, yeast extract, malt extract, ground banana, orange juice and tomato juice are added to supplement some basic media, since they have been reported to enhance the growth of cultures. The next component are solidifying agents. Plant tissue culture media may be solid or liquid depending upon the presence or absence of a gelling or solidifying agent in the medium. Agar, which is a polysaccharide obtained from seaweeds at concentrations ranging from 0.8 to 1%, is the most commonly used gelling agent for preparing semi-solid and solid plant tissue culture media. Liquid medium is used for suspension cultures, while a semi-solid or solid medium is used for culturing plant tissues on the surface of the medium. The next component is growth regulators. The main classes of plant growth regulators and their role in plant tissue culture are: first, auxins. Auxins promote both cell division and cell growth, and common auxins used in plant tissue culture media include indole 3-acetic acid or IAA, indole 3-butyric acid or IBA, 2,4-dichlorophenoxyacetic acid or 2,4-D, and naphthalene acetic acid or NAA. The second group of growth regulators are the cytokinins. Cytokinins stimulate cell division, induce shoot formation and axillary shoot proliferation, and retard root formation. While the ratio of auxins to cytokinins control morphogenesis in plant cell cultures in the following way. A ratio of high cytokinin to low auxin leads to regeneration of shoot part, while low cytokinin to high auxin leads to regeneration of root parts. Next, medium cytokinin to medium auxin ratio leads to regeneration of both shoot and root. And finally, medium cytokinin to low auxin ratio leads to callus formation. Cytokinins commonly used in culture media include BAP, that is 6-benzyl-aminopurine, 2-IP or 6-dimethylaminopurin, kinetin, zeatin, and TDZ or thidiazurin. The next group of growth regulators are the gibberellins. Gibberellins enhances growth of callus and help in elongation of dwarf plantlets, and GA3 is the most commonly used gibberellins in plant tissue culture. The next growth regulator is abscisic acid or ABA. ABA is used in plant tissue culture to promote some distinct developmental pathways such as somatic embryogenesis. Then, the last group of plant growth regulator is ethylene, which is a naturally occurring gaseous plant growth regulator controlling fruit ripening in climacteric fruits and is not commonly used in plant tissue culture. The second factor affecting cultured plant tissues is explant source. Explants are the small parts or segments of plant tissues which are cultured on cultured media to initiate cultures. Plant segments commonly used as explants in tissue culture include stem, root, leaf, flower, ovule, cotyledon and hypocotyl, thin cell layer, embryos, shoot tips and meristems, etc. The source of explants is extremely important for the success of tissue culture studies, and the factors affecting explants tissue culture response are: first, genotype. The in vitro culture response of cultured plant tissues varies widely among families, species, and even within genotypes from the same species. Generally, the regeneration capacity of dicotyledons is more than monocots, and also herbaceous plants regenerate more easily than woody plants such as trees and shrubs. Further, among the dicotyledons, plants from some families such as Solanaceae, Cruciferae, Begoniaceae and Crassulaceae etc. have high regeneration capacity, and transformations are quite easy in members of these families, while plants belonging to some family may be recalcitrant with respect to in vitro culture and genetic transformation. The second factor is the physiological stage of donor plant. Generally, explants isolated from young tissues and organs or vegetative segments have high regeneration capacity than those from mature and generative tissues of plants. Moreover, explants isolated during flowering period regenerate more easily in vitro than during resting or dormant stage. The third factor is the source of explant tissues. Tissue culture response of the explants used for starting culture is also affected by the weather conditions of the place where the stock plant is grown. In general, explants isolated from in vitro grown seedlings give the best response, followed by plants grown under greenhouse conditions and the ones grown in field conditions. The fourth factor is the explant age. The regeneration capacity of explants decreases with increasing physiological age of the plant organ used as explants for in vitro organogenesis. Next factor is the size of explant. Large explant such as leaves and hypocotyls consisting of parenchyma, vascular tissues and cambium, or explants having a big amount of nutrition reserves such as tubers and bulbs have greater regenerative capacity compared to smaller explants such as sails and meristems due to limited nutrients and hormone reserve in small explants. The next factor is the explant position in donor plant. The response of explants in tissue culture may also vary depending on the place from where the explants are excised. The next factor is explant density. The density of explants on the culture medium also influences the explant's regeneration capacity, since the explants compete with each other for a constant amount of water and nutrients in growth medium. Let us now take up the third factor, that is the culture environment. The culture vessels containing explants placed on growth medium are kept in culture rooms where the environmental factors such as temperature and light are controlled. Some of the general environmental factors affecting in vitro cultures are: first, lighting. Normally, organogenesis in culture takes place with an illumination of 2000 to 3000 lux provided by fluorescent tubes with a 16-hour light and 8-hour dark photoperiod. Moreover, the blue region of spectrum promotes soot formation and red light induces rooting, while the treatment of blue light followed by treatment of red light also stimulates organogenesis. The second condition is the temperature. The temperature in culture room is usually controlled, allowing variation of only about 25 plus minus 1 degree Celsius, as changing temperature regime causes stress in cultures which inhibits growth. The third factor is the oxygen gradient inside the culture vessels. Low gradient of available oxygen inside the culture vessel favors soot bud formation in some cultures, while rooting requires a high oxygen gradient. The final condition is the pH of the medium. Optimum pH range from 5 to 6 is suitable for in vitro growth of explants, and it affects the uptake of ions by the explants. Usually, the pH higher than 6 results in a fairly hard medium, whereas pH below 5 does not allow satisfactory solidification of the medium. The pH of the medium generally falls by 0.3 to 0.5 units after autoclaving. Now let us see the conclusion. Tissue culture studies generally aim at obtaining high-frequency shoot regeneration, which is also a prerequisite for an efficient transformation system and clonal propagation of plants. However, the optimum growth and morphogenesis of the plant tissues cultured under sterile conditions is affected by many factors like the growth media, explant use, and the culture environment, etc. And determining the factors affecting regeneration capacity of explants will increase the success of tissue culture studies, since every cell has an ability of forming a whole fertile plant under in vitro conditions. Thank you.